My name is Ahmad Khalil. I'm an assistant professor in genetics and genome sciences. Today we will be talking about a novel set of genes in the human genome that we refer to as lincRNAs. A snapshot of the modern view of the human transcriptome. Now we know that there's approximately 22,000 protein-coding genes in the human genome. That only constitutes about 2% of the entire human genome. We also know that there is a plethora of different types of non-coding transcripts that are encoded within our genome. This includes classic non-coding RNAs such as ribosomal RNAs and transfer RNAs, and others that are required for splicing and protein synthesis. What we will be focusing more on today is regulatory non-coding RNAs. This includes microRNAs, which we have about 1,000 of them in the human genome. piRNAs, those are germ cell-specific types of RNAs. They are not expressed in somatic cells. And lastly, a novel and a new exciting class of non-coding RNAs that we refer to as lincRNAs. And there's approximately 10 to 15,000 of those transcripts in the human genome, and the number may increase over time. We previously identified human lincRNAs using histone modifications marks of active genes. Specifically, we took advantage of specific histone modifications that are found in the promoter region and in the body of the gene, and eliminated any known protein-coding genes that corresponded to these histone modifications, and we focused on novel intergenic regions that have these histone modification patterns as shown, as circled here in red. Using this method, we identified over 3,000 of these novel lincRNAs in 6 human cell types. However, when other investigators looked in other tissues and cell types, they were able to identify additional lincRNAs. The number of lincRNAs in the human genome are now estimated to be over 10,000. If we look at the cellular localization of these human lincRNAs, what you are looking at in blue is the nuclei of cells, and the white dots correspond to the hybridization of a specific probe for each one of these lincRNAs. For example, the lincRNA exists, which is expressed in female somatic cells, codes the inactive X chromosome. It's not present in a male cell line, as there's no X inactivation in males. These other novel lincRNAs show different types of cellular localization. For example, this particular lincRNA over here shows stronger nuclear localization. This is in contrast, for example, to TUG1, which shows both nuclear localization as well as many cytoplasmic localization. So it might have both a nuclear and cytoplasmic function, while other lincRNAs may have exclusively nuclear function. We were interested in the function and mechanisms of these lincRNAs. We use a technology called RIP-seq to potentially identify interactions between lincRNAs and epigenetic-modifying complexes. This has included PRC2. We identified genome-wide interactions between PRC2 and a subset of lincRNAs. More recently, we looked at interactions between DNMT1, the DNA methyltransferase, and lincRNAs, and we also were able to identify a subset of lincRNAs that specifically associate with DNMT1. So these interactions suggested that these lincRNAs could be mediating epigenetic mechanisms of gene regulation in the nucleus. Based on these findings, we proposed a model where these lincRNAs are transcribed by RNA polymerase II, and they form some form of secondary structure that we don't completely understand yet. This secondary structure allows them to interact with these epigenetic complexes such as PRC2 and guide these complexes to specific sites of the genome where PRC2 now can modify chromatin, in this case adding lysine 27 trimethylation to repress gene expression. This mechanism offers a great opportunity for therapeutic intervention. For example, if we can block the interaction between PRC2 and a specific lincRNA, we can activate specific gene expression in the genome. This is in contrast to completely knocking down components of PRC2, where that would lead to the activation of thousands or hundreds of genes. A key thing that we have learned recently that cancer is a genetic as well as an epigenetic disease. There are numerous epigenetic changes that occur in tumors that we now believe lead to the progression of carcinogenesis, or potentially the initiation or the metastasis of these tumors. One key epigenetic modification that occurs in cancer is loss of global DNA methylation. So in cancer, there is a global loss of DNA methylation. Keeping in mind there is increased methylation at specific sites in the promoters of tumor suppressors, but for the most part, the cancer epigenome is hypomethylated. However, the mechanisms that lead to this hypomethylation are not known. We know that this hypomethylation causes changes in gene expression. It causes genomic instability. And we know it causes aggressiveness of these tumors. So we postulated that there is the potential for some of these lincRNAs that interact with these enzymes that mediate DNA methylation, and that the deregulation of these lincRNAs might cause changes in DNA methylation without changes in the expression of these methyltransferases themselves. And we focus on DNMT1 since it's a key enzyme involved in DNA methylation. As I discussed earlier, we found that DNMT1 interacts with a subset of these lincRNAs, and we selected a top candidate of these lincRNAs for functional studies to determine if that affects DNMT1 function. This particular lincRNA that we selected is expressed in normal colon tissues, shown in blue. However, it's repressed in a large panel of colon cancer patient-derived cell lines, shown in red. And as you can see, some of these cell lines have very low expression of this particular lincRNA. What happens if we put this lincRNA back into these cells? Would the DNA methylation be restored in these cells? And that's exactly what we observed in two separate cell lines. And what we found that when we re-express DACOR1 in these cells, we gain DNA methylation at numerous sites in these cells, suggesting that now DNMT1 is able to go back to these sites and methylate DNA. Also, we observed that when we re-express DACOR1 in these cells, it slows the growth of these cells. These are three different colon cancer cell lines. And as you can see, when we put DACOR1 back in, again, it slows the growth of these cells, in some cases very substantially. So it suggests this is a growth suppressor function for this lincRNA. To summarize how this particular lincRNA works, and hopefully gives you a framework for other lincRNAs that could potentially work through epigenetic mechanisms. In colon cancer cells, this particular lincRNA becomes repressed. This leads to upregulation of genes involved in metabolism. For example, these genes are involved in serine biosynthesis, which increases PKM2 activity. Also, the upregulation of CBS results in reduction of a key substrate called S-adenosylmethionine, which is required for DNA methylation. The loss of SAM in these cells leads to loss of DNA methylation and causes genomic instability. Just to summarize the key points from this presentation, we have demonstrated through this study and other studies that the dysregulation of lincRNAs is a major contributor to epigenetic alterations in cancer cells. These studies are a paradigm shift in our understanding of gene regulation and dysregulation in cancer and how that contribute to the tumor genesis. These lincRNAs, they are very novel, and they open the door for RNA-based therapeutics where we can target the lincRNAs in these tumors. One advantage of targeting lincRNAs is that they are very tissue-specific in their expression, and that could potentially reduce side effects in clinical treatments of different cancers. Finally, I just want to remind you that not all lincRNAs are involved in epigenetic regulation. LincRNAs have many diverse roles in cells, and they can work in the nucleus or the cytoplasm by various mechanisms.